Welcome back to the Deep Dive. Today we are looking at a story that, honestly, it involves a perilous migration, a high-stakes obstacle course, and a survival rate that, well, it's shocking when you actually look at the math.
It really is. And,
Yeah.
we aren't talking about some nature documentary about salmon swimming upstream or anything like that. We're talking about us, every single one of us.
We're talking about Gametogenesis
Mhm.
and the first week of development. This is basically the prequel to being a person. And we've got the heavy hitters on the desk today. Langman's medical embryology, Morin Prasad's The Developing Human, and I.B. Singh's human embryology.
The holy trinity for any first semester medical student. I mean, these texts are dense, they are incredibly detailed, and they are, without a doubt, the gold standard.
Exactly. And our mission today is to take these dense, diagram-heavy chapters, specifically the ones on Gametogenesis and the first week, and really decode them. We want to find the story, the narrative that's kind of hidden in all the jargon. Because when you actually read Langman, for example, you realize the cells that make us, well, they don't start where you think they do.
That is the first big surprise, isn't it? It's something we're calling the wanderer concept.
Right. And before we get into the weeds, I want to just hit you, the listener, with the big takeaways we're going to cover. So we're going to talk about that wanderer. We're going to unpack the great asymmetry between how men and women make gametes. It's fundamentally different.
We're also going to break down the week of twos, which is a classic mnemonic, a great memory aid to help you remember the structure of that first week. And then we have to face the clinical reality, the cold, hard math of just how many conceptions actually make it.
It's surprisingly low. And just a quick disclaimer before we launch, we are synthesizing these texts. We're trying to give you those aha moments, that big picture. This is a revision aid and hopefully a way to spark your curiosity.
Yeah.
It is definitely not a replacement for cracking open those books and really looking at the diagrams yourself.
Absolutely not. Nothing replaces the primary text. Think of this as the trailer that makes you really want to go and watch the movie.
Yeah. Okay, let's get into it. I want to start with the prequel, because I think if you stop someone on the street and ask, hey, where do sperm and eggs come from? They'd probably look at you a bit funny and then say, well, the testicles and the ovaries.
Which is, a perfectly logical answer. It just happens to be embryologically completely wrong.
It is. And I.B. Singh is really, really clear on this. These cells, the primordial germ cells, or PGCs for short, they are essentially nomads. They do not originate in the gonads. They originate way back in the second week of development in a layer called the epiblast.
Right. And to help everyone visualize this, because the embryo at this point is basically a tiny flat disc, these cells actually move out of the embryo entirely. They migrate through a structure called the primitive streak, and then they set up camp in the wall of the yolk sac.
The yolk sac. So they're hanging out in what is basically a nutrient pouch outside the main developing body. And I was reading through more, trying to figure out why. Why would they go all the way out there just to come back later? It just seems so inefficient.
It really does seem that way, doesn't it? But there's a method to the madness. The leading theory is all about protection. The embryo proper is this hotbed of signaling molecules telling cells you become skin, or you become muscle, or you become a nerve.
It's like a construction site with orders being shouted everywhere.
Exactly. And you absolutely do not want your master files, the germ cells, to get corrupted by those instructions. You need them to stay pluripotent. They have to remain blank slates. So nature sequesters them. It hides them away in the yolk sac, far from all that chaos, until it's safe to bring them back home.
But that return journey, that's where the real danger lies, right?
Yeah.
I.B. Singh describes their movement as amoeboid. They're literally crawling cells.
It's a physical hike. It's an incredible trek. Around the fourth week, they finally leave the yolk sac. They start crawling along the wall of the hindgut. Then they move through the dorsal mesentery. That's the tissue that suspends the gut. And they were aiming for these two ridges of tissue, the genital ridges.
Which are the primitive gonads, the structures that will one day become the testes or ovaries. So effectively, your future children are crawling up your own back wall while you're still an embryo.
That is a very strange way to put it, but it's completely accurate. And Langman adds a really important detail here. They aren't just walking, they're multiplying as they go. Mitotic divisions are happening during this migration. They're building an army while they're on the march.
But here's the thing that really stood out to me across all the texts. This isn't a straight line. It's a biological obstacle course. And sometimes they get lost.
And this is where we get one of the most fascinating and, frankly, disturbing clinical vignettes in the entire book.
The sacrococcygeal teratoma.
It's a mouthful, for sure.
It really is. But picture this scenario. a newborn baby girl, otherwise totally healthy, but she presents with this huge tumor, a mass at the base of her spine. Langman says this is the most common tumor in newborns, occurring in about 1 in 20,000 to 70,000 births.
And the origin of this tumor is those lost wanderers. These are PGCs that they strayed from the path. They got lost and settled in the coccygeal region instead of making it all the way to the gonads.
And because they are, as you said, these master files that can become absolutely anything, they don't just form a simple lump of cancer cells. They actually try to build a person.
That's the part that just catches everyone off guard. These tumors are pluripotent.
Which means they can contain teeth, they can contain hair, muscle, bone. Singh even mentions they can contain organized structures like loops of gut or even, fingers and toes.
It's sometimes called the monster tumor, but it serves as this incredibly powerful proof of concept. It proves beyond a shadow of a doubt that these microscopic cells really did hold the potential to create an entire human being. They just lacked the direction, the regulatory environment of the gonad to do it properly.
It's a really stark reminder of how controlled and precise normal development has to be.
Okay, so let's assume the wanderers actually make it. They arrive at the genital ridges, they set up camp. Now we enter the phase called gametogenesis.
Right, so this is where we convert these raw germ cells into the actual players, the sperm and the egg. And the goal here is really twofold: meiosis and cytodifferentiation.
Meiosis is the math part. That's about reducing the chromosomes from 46, our normal diploid number, down to 23, the haploid number. You need half from mom, half from dad. Simple.
And cytodifferentiation is just a fancy way of saying changing shape. But this is where we see what we call the great asymmetry. The way males and females handle this whole process is completely different. It really is like the tortoise and the hare.
Okay, let's look at the male side first. I.B. Singh describes it as a factory model. But there's a crucial detail in the anatomy of this factory that I think a lot of people miss: the security system.
Ah, yes. You're talking about the blood-testis barrier. Yes. This just blew my mind. So we know that the spermatogonia, the stem cells, they sit at the base of the seminiferous tubules. But as they start to divide and change into sperm, they have to cross a physical barrier created by these other cells, the Sertoli cells.
And that barrier is absolutely vital, because here is the biological reality. Sperm are genetically different from the man who produces them. They have half the chromosomes. They've undergone recombination, so the genes are all mixed up.
So to the body's immune system, a sperm cell looks like an invader. It looks like a virus or a bacterium. It's non-self.
Exactly. If your immune cells could see your sperm, they would attack them immediately. It would be a massive autoimmune reaction. So the Sertoli cells literally link arms. They form these things called tight junctions to create a completely sealed off environment. The sperm are developed in a safe room, hidden from the body's police force.
That is just incredible. The body has to hide its own reproductive cells from itself. And inside that safe room, the transformation is pretty dramatic. We aren't just splitting cells, we're engineering a vehicle.
That's spermiogenesis. It's the difference between a raw block of steel and a Ferrari. You start with this round, clumsy cell, the spermatid. It's heavy, it's immobile, it's useless for fertilization.
And Langman breaks down the renovations really well. First, you have to strip the weight.
Right. The nuclear condenses down. You pack the DNA as tight as physics will allow. The Golgi apparatus, which usually just handles protein packaging, it transforms into the acrosome.
The chemical helmet.
A helmet filled with enzymes. It covers the front half of the nucleus because it's going to have to drill through the egg's defenses later on.
And the engine. What about the engine?
That would be the mitochondria. Now, in a normal cell, mitochondria just kind of float around everywhere. But in a sperm, they all migrate. They wrap themselves in this tight spiral around the middle piece of the tail. It's literally designed like an outboard motor.
All to power that flagellum, that tail.
Mm-hmm.
But even then, Singh notes that when they leave the factory, when they actually leave the testes, they can't really swim yet, can they?
No, they can't. They're structurally complete, but they're functionally immature. They're just pushed along passively into the epididymis. They need time to mature there, to learn how to swim, essentially. The whole process takes about 64 to 74 days from start to finish.
Which is why, clinically, if a man has a high fever or takes a certain drug that affects sperm production today, you might not see the result of that in a fertility analysis for almost 3 months.
Exactly. You're always looking at a snapshot of what was happening in the factory 2 or 3 months ago.
Okay. Now, let's contrast all of that with the female process. So if the male is a factory running 247 from puberty until death, the female system is. It's like a vault.
A vault with a timer, a very long one. According to Langman and Moore, in females, the entire process of generating germ cells happens before birth.
Wait, let's just pause on that. A female fetus, still in the womb, already has all of the eggs she will ever have for her entire life.
Yes. And not only that, but by the 5th month of fetal life, she has about 7 million of them. That is the peak number she will ever have. But then a massive die-off begins, a process called atresia. By the time she is born, millions are already gone. By the time she reaches puberty, she's down to about 40,000.
From 7 million down to 40,000, that is a staggering loss.
It is. And of those 40,000, only about 500 will ever actually be ovulated during her entire reproductive life.
So unlike the male factory model, the female system is all about selection, conservation, and very, very long periods of waiting.
And the waiting is chemically enforced. The primary oocytes, they enter meiosis this first, and then they hit a pause button. They stop in a specific stage of prophase called the dichiotin stage. And they stay frozen there for 15, 30, maybe even 45 years.
That is just wild to think about. A cell sitting in suspended animation, perfectly preserved for 4 decades. What wakes them up?
Puberty. And then each month, the LH surge, that's luteinizing hormone, it stimulates a small cohort of them to finish that first meiotic division. But here's another key asymmetry. When a male cell divides, it makes 4 equal sperm. When a female cell divides, it makes 1 giant egg and 1 tiny little polar body.
And the polar body is basically just a trash bin for the extra chromosomes, right, to get down to 23.
That's all it is. The egg needs to hoard all the cytoplasm, all the nutrients and machinery, because it's going to have to run the entire show for the first few days of embryonic life. It can't afford to split its resources, so it just dumps the extra DNA into a polar body, which eventually just degenerates and disappears.
And then it hits another pause button.
Yes. The secondary oocyte starts meiosis the second, but it arrests again in metaphase Tipkin, and it will only finish that division if it is fertilized by a sperm. If no sperm shows up, it never completes meiosis.
So technically, an unfertilized egg is a cell that never actually fulfilled its cellular destiny. It just dies in a state of arrest.
Precisely. And this long waiting game, this extended dichiotin stage, it has very serious clinical consequences. And that brings us to our next section. What happens when division goes wrong?
We're talking about non-disjunction.
Yes. Non-disjunction is simply the failure of chromosomes to separate properly during meiosis. And as Langman points out very clearly, the biggest risk factor for this is increasing maternal age.
Because the machinery gets rusty.
That's the colloquial way to put it, and it's actually pretty accurate. The protein machinery, the spindle fibers that are supposed to pull the chromosomes apart, has been sitting dormant in that dichiotin arrest for 40 years in some cases. It can degrade, it can become less efficient, so when the signal finally comes to separate the chromosomes, sometimes they stick together.
And this leads to the syndromes that we all recognize from medical school.
Trisomy 21 or Down syndrome is the most well-known example. That's where you have an extra copy of chromosome 21, and it leads to features like micrognathia, a small jaw, and that characteristic broad face. But there are many others.
Like Turner syndrome.
Turner syndrome is fascinating because it's a monosomy. Genetically, it's 45X, and listeners should really note this. It is the only monosomy that is compatible with life. These are females who present with short stature, a webbed neck, and no ovaries, what's called gonadal dysgenesis. But even then, about 98% of all Turner fetuses are spontaneously aborted.
And that leads us to the statistic that it blew my mind the first time I read it in Moore and Prasad. The statistic that 50% of all conceptions end in spontaneous abortion.
It is a shocking number, 50%, half. And about half of those are due to major chromosomal anomalies like the ones we're talking about.
So in a way, miscarriage isn't a failure. It's a natural quality control mechanism.
That is exactly how the texts frame it. It is a biological screen. If the chromosomal math doesn't add up correctly, development simply halts. It really explains why that early period of pregnancy is so incredibly fragile.
Okay, let's pivot from there. Let's assume the gametes are healthy. We have a good sperm, we have a good egg. Now we get to the main event, fertilization.
And this is not a simple meeting. It's an obstacle course. The sperm has to travel all the way from the vagina, up through the uterus, and into the ampulla of the uterine tube. That is the normal site of fertilization.
And I.B. Singh has a specific box about this. I think it's box 2.2 on capacitation. Even if a sperm swims all the way there, it can't fertilize an egg right away, can it?
No, it can't. Freshly ejaculated sperm are chemically blocked. They need a conditioning period. It takes about 7 hours in the female reproductive tract, and this process is called capacitation.
I always think of this as the sperm having to strip off its armor before battle.
That's a great way to think about it. They lose a glycoprotein coat and some seminal proteins that are covering the acrosome. This makes the membrane more fluid and it becomes hyperactive, ready for action. Without capacitation, the sperm is sterile.
So the sperm is capacitated, it finds the egg, now it has to break in.
Right. Step 1. Penetrate the corona radiata. These are the follicular cells still clinging to the outside of the egg. For this part, the sperm basically just uses brute force and its swimming speed.
And step 2.
The zona pellucida. This is the egg's tough glycoprotein shell. This requires the acrosome reaction. The sperm's chemical helmet bursts open and releases enzymes like acrosin to literally bore a hole through that shell.
And once it touches the egg's actual membrane,
Fusion. The two plasma membranes merge. But this raises a really important question. What stops 100 other sperm from getting in at the same time? Because that would be disastrous.
that's polyspermy.
Right. If more than one sperm gets in, you have too much DNA. The whole thing is non-viable. The cell dies. So the egg has a brilliant defense mechanism. The very instant the first sperm fuses, the egg undergoes the cortical reaction.
What happens there?
The egg releases these cortical granules right beneath its membrane. The contents of those granules spread out and chemically harden the zona pellucida. It's like slams the door shut and throws the deadbolt. Within seconds, it makes the shell completely impenetrable to any other sperm. This ensures that the diploid number is restored to exactly 46 chromosomes. It also, of course, determines the sex of the embryo X or Y from the father, and it provides the metabolic kick to initiate cleavage. The egg wakes up.
Okay, so we have a zygote. Day 1. Now begins the first week of development. And the texts all describe this as the journey to the uterus.
And here's where we need to introduce that week of twos pattern. But before we get to the twos, we really have to talk about the paradox of cleavage.
The paradox being that the cells divide, but the embryo doesn't actually grow.
Exactly. The zygote divides into 2, then 4, then 8 cells, and these cells are called blastomeres. But you have to remember, it is still trapped inside that hardened zona pellucida shell. It physically cannot get bigger, so the cells just get smaller and smaller with each division.
It's like slicing a pizza into more and more pieces. You have more slices, but you still have the exact same amount of pizza.
That's a perfect analogy. But then around day 3, something really crucial happens. It's a process called compaction.
This is the group egg, right?
Yes. Both Langman and Moore really emphasize this point. Up until the 8-cell stage, the cells are just this loose arrangement. But then suddenly, they maximize their contact with each other. They squeeze together tightly and form gap junctions. And this is the first time the cells really start talking to each other and decide who is going to be what.
So this is the first real step of differentiation.
It is. They segregate into 2 distinct populations, an inner cell mass and an outer cell mass.
And the inner cell mass is destined to become the baby.
Right. And the outer cell mass is going to become the placenta and the supporting membranes. Langman even gets into the molecular mechanism here. It's the hippo signaling pathway that is responsible for telling the cells which group to join.
So now we have this solid ball of cells called the morula, I guess because it looks like a mulberry.
Exactly. And that morula enters the uterine cavity around day 3 or day 4. Then uterine fluid starts to seep inside, creating a cavity. Now we have to change the name again. It is officially a blastocyst.
And this is where the hatching happens. I love this visual.
It is absolutely critical. The embryo is still inside that hard shell, the zona pellucida, but in order to attach to the wall of the uterus, it has to be sticky. The shell isn't sticky. So the blastocyst literally has to burst out of the zona pellucida. It hatches like a chick from an egg.
And if it doesn't hatch, it can't implant. Game over.
Correct. And implantation begins around day 6.
Day 6. This is when the rolling tennis ball slows down.
It's a great analogy from Langman. The trophoblast cells, that outer layer, they express these receptors called L-selectins that catch onto the uterine wall. It's just like a leukocyte slowing down in a blood vessel or a tennis ball rolling to a stop on a patch of Velcro. It's an adhesion process.
And once it sticks, it invades. And this is where the week of twos really kicks in.
Yes. So as the trophoblast invades the uterine wall, it differentiates into 2 distinct layers. You have the inner cytoprophoblast, which is a layer of distinct individual cells, and then you have the syncytiotrophoblast, the outer, incredibly aggressive layer.
Now, syncytio means joined, right? Like a syncytium.
Yes. It loses its cell boundaries. It becomes this single, giant, multinucleated mass of cytoplasm that acts like a root system, just burrowing deeper and deeper into the mother's endometrium to tap into her blood vessels for nutrients. It is incredibly aggressive, invasive behavior.
And if that kind of behavior happened anywhere else in the adult body,
We would call it cancer. And that's not just a casual comparison. Biologically, the molecular similarities between early implantation and tumor invasion are striking. To survive, the early embryo acts like a controlled tumor.
That is a very sobering thought. We have to talk about how this textbook biology is actually used in the real world. Because for millions of people, this natural journey we just described just doesn't work.
And that's where we enter the modern era of assisted reproductive technology, or RT.
Right. Moore and Prasad have a whole section on this.
Yeah.
We're talking about IVF. But I want to zoom in on something specific that relates back to that cleavage phase we discussed. The idea that these early embryonic cells are totipotent.
This is the entire basis of pre-implantation genetic diagnosis, or PGD.
Okay, so imagine you're in an IVF lab. You have an embryo in a dish. It's day 3. It's just 8 little cells inside that zona pellucida shell.
It just looks like a microscopic blackberry under the microscope.
Right. Now, to check if that embryo have a specific genetic disease, let's say cystic fibrosis, the embryologist can actually reach in with a microscopic pipette and remove one of those 8 cells.
They literally subtract 1/8 of the organism.
And the crazy thing is the embryo doesn't care. The remaining 7 cells just reshuffle, compensate, and go on to build a perfectly normal human being.
It really highlights the incredible regulative capacity of the early embryo. The cells haven't really decided their final fate yet. They're still flexible.
But this leads to the ethical question that Langman sort of touches on, but I think we should push on it a bit. If we can screen for cystic fibrosis, well, we can screen for other things.
Of course. We can screen for sex. We can screen for chromosomal aneuploidy, like Down syndrome.
And as we get better and better at reading the entire genome, where do we draw the line? I mean, we talked earlier about how 50% of conceptions fail naturally. That's nature's quality control. But now we are the quality control.
We are absolutely stepping into the driver's seat. You know, in the past, a sperm with poor motility just wouldn't fertilize an egg. That genetic line would naturally end. But now, with a technology like ICSI, intracytoplasmic sperm injection, we can take that single sperm that couldn't even swim, pick it up with a needle, and inject it directly into the egg.
We're completely bypassing the obstacle course.
We are. And on an individual level, that is a miracle. It allows a couple who couldn't otherwise to have a child. But on a population level, from a cold evolutionary biology standpoint, we are retaining genes in the gene pool that nature was actively trying to filter out.
That's a really heavy thought. Are we fundamentally changing the definition of biological fitness for our own species?
I think we undoubtedly are. We just won't know the consequences of those changes for a few hundred years.
It's the ultimate trade-off, isn't it? Between compassion for the patient sitting in front of you today and the unknown future of the human genome.
And it all starts right here in that first week.
So let's wrap this up. We've gone all the way from these wandering primordial germ cells to the high-tech, high-stakes IVF lab.
It's quite a journey in just 7 days.
I want to leave the listener with that week or two summary again, just to help lock it in.
Sure. So by the end of the first week, the trophoblast has differentiated into 2 layers, the cytotrophoblast and the syncytiotrophoblast. And at the same time, the embryoblast is organizing itself into 2 layers, the epiblast and the hypoblast.
Which is a perfect foreshadowing for next week's deep dive into the bilaminar disc.
Exactly. The story continues.
Okay, here is my final thought for you, the listener. Do yourself a favor. Open your textbook, whether it's Langman or Moore or Singh to the first week chapter. Find that diagram of the blastocyst hatching from the zona pellucida.
It's a beautiful image. It's the moment the embryo decides to engage with the outside world.
It is. And it's the first time you can visually verify the difference between what will become the baby and what will become its life support system. Thanks for listening to The Deep Dive. We'll catch you next time.